Mikrofon. Thank you, Paolo. Giriş için teşekkürler Tuğrul Bey. Tüm katılımcılarımıza eğitim ve seminerlerimize verdiğimiz uzun bir aradan sonra ben de hoş geldiniz diyorum. Bu açığı 2025'te kapatacağımızın müjdesini de verebilirim herhalde. Mikrofonları açık olan katılımcılarımız var. Onları uyaralım. İlaç ve tıbbi cihaz sektörüne test hizmetleri veren iş ortağımız EUROFINS'in kıdemli danışmanı olan Paolo webinarı İngilizce sunacak. Webinar sonunda kendi ürünlerinize ait spesifik sorular dahil olmak üzere tüm sorularınızı Paolo'ya yazılı veya sözlü, ister İngilizce ister Türkçe yönlendirebilirsiniz. Elimizden geldiğince çeviri konusunda Türkçe sorularınıza yardımcı olacağız. After a short welcome in Turkish, we are going to continue the rest in English. At the end of the presentation, we plan a 30-minute question and answer section where you may ask your questions through chat box or by activating your microphone. It's a pleasure for us to host you again with a new topic, Paolo. Will you please give a short introduction about EUROFINS and yourself before the webinar?
Yes, sure. And thanks, Tunka, for inviting me, and thanks to everybody here. I see a lot of participants. We are close to 200 participants. That's great. So I'm Paolo Pescio. I'm a Senior Scientific Director at EUROFINS Medical Devices Service Europe. In EUROFINS, we provide a service for medical device manufacturer, both in terms of testing, sterile packaging, and consultancy. And today, I'm going to share with you something about toxicological risk assessment of medical device, one of the services that we do provide to our clients. Before starting, just a few words about me. I'm a biomedical engineer with a background mainly in biological evaluation and toxicological risk assessment, and I had the chance to be involved in biological evaluation of medical device for the last almost 20 years. And since 2009, I'm an Italian expert for ISO TC 194 and SEN TC 206 that are the two technical committee involved in biological evaluation of medical device, mainly related to the 10A3 series. I'm also convener of working group 2 of SEN 206, that is the working group with the focus on NextZ, so with the focus on the harmonization of the 10A3 series under the MDR. Having said that, I like to share my screen. It should work. If it's not the case, just let me know, but it seems working. Sorry, when on sharing the screen, I cannot read your question live, but as Tunka said, we have a 30 minutes Q&A at the end of my presentation. And I hope to stay in less than 1 hour to accommodate your question. Of course, if we have an answer question, I will follow up by email, or if you like to send your question later on by email, please do not hesitate. I will again follow up by email. So today I'm going to share with you something about an 10A3-17 of 2023, a new standard that was published in September last year, and it's about toxicological risk assessment for medical device. What I would like to share with you today is my experience with this standard, and generally speaking with toxicological risk assessment of a medical device. Okay. Every time we speak about toxicological risk assessment, we have to cite Paracelsus. It was, as you know, a Swiss physician, alchemist, and a philosopher, and he is credited as the father of toxicology. He said, once all things are poison and nothing is without poison, the dose alone make it. So a thing is not a poison. It's indeed a matter of dose. So we are really interested in the dose the patient is exposed to. May I kindly ask to mute your mic because there is an echo here? Thanks a lot. We'll see today how to apply this concept, the dose, and how to determine the dose the patient is exposed to the assessment of constituent of medical device. But why we are so interested in constituent of a medical device? Because from ISO 103-1, we know that the chemical characterization is a prerequisite for the entire biological evaluation. And the characterization shall address the constituent chemicals of the medical device and possible residual process aids or additives used in its manufacture. So we are really interested in knowing the chemicals the patient is exposed to. And we have two definitions of constituent, or at least I have to say we have three. Let's start with the first two definitions. One definition is coming from 103-1, says that a constituent is any synthetic or natural substance that is used in a process for manufacturing materials or medical device, including the base materials, additives such as antioxidant, UV stabilizer, color additives, dye, and so on, and processing aids. So this is the definition from part one with a clear focus on what is intentionally added to the medical device. It's a list of raw material plus additives used during the manufacturing process. While the definition of constituent according to part 18 is slightly different, but there is one added part that is particularly crucial in our assessment. The 1093-18 included the concept of impurity, so something that is not intentionally present in the medical device. The definition is that a constituent is a chemical that is present in a finished medical device or in its material of construction, that it may be intentionally present, additives, antioxidant, and so on, like the definition in part one, or unintentionally present, like an impurity. So we have this addition according to part 18. But I say that we have three definitions because in September last year we have this new definition coming from 1093-17. A chemical says that a constituent is a chemical that is present in or on the finished medical device or its material of construction. So there is this specification that the chemical can be present also on the surface of the medical device, and the rest is the same as in part 18. So something that can be intentionally or unintentionally added to the medical device. So the definition of part 18 was modified to make clear that a constituent can be also on the device. A patient, let me say, doesn't care if he or she is exposed to a constituent in the medical device or on the medical device. The patient is interested in the dose he is exposed. And in our assessment, in our chemical characterization, we should take into account the constituents that are in or on the medical device. And to be honest, when we extract the device in order to perform, for example, an extractable study during the chemical characterization, we extract everything that is inside the device and everything that is on the device, on the surface. I say that we can perform an extractable when we are performing a chemical characterization, because I want to highlight here that a chemical characterization is a process for obtaining chemical information. Chemical characterization is not only an extractable study, or it's not only a leachable study. Is really a process, is quite a complex process. And the flow chart guiding the chemical characterization process is the one, or the process, the flow chart is just the representation of our process, is the one provided in 1093-18. where, as you know, we find a question repeated 3 times in a different stage of the process. The question is, does risk assessment conclude
Paolo, excuse me. Could you open
Paolo,
up your
could you please stop,
please?
because we can't hear you.
Microphone.
Okay, can you hear me? I haven't unmuted the
Yes.
mic, so
Yeah, we can.
where did you experience issue?
At the beginning of this slide, does
At the
risk
beginning.
assessment conclude
Okay,
device has
okay.
acceptable risks?
Perfect. Thanks a lot. Sorry for that.
You're
Probably people entering and muting the micro. Okay, I hope that it's we are not experiencing anymore these issues. Sorry for that, and thanks for letting me know. So as I said, I try to say it again. So the chemical characterization is a process. The process for the chemical characterization is described in ISO 1083-18, and here we have a representation of the process of this chemical characterization through a flow chart. In this flow chart, as you know, there is a question repeated three times that says, does risk assessment conclude device as an acceptable risk? So we need to answer to this question at different stage of the process. And in order to answer to this question, we are asked to perform a toxicological risk assessment. So the toxicological risk assessment is an essential part of the entire chemical characterization, and we need to perform a toxicological risk assessment after the compositioning profile stage. So the stage that you see here, that is the compositioning profile stage. Then we have this question, or after the extractable assessment, we have the very same question, or after the leachable step. Again, the toxicological risk assessment. So really, the toxicological risk assessment is an essential stage for the entire chemical characterization. The toxicological risk assessment is defined as the act of determining the potential of a chemical to elicit an adverse effect based on a specific level of exposure. So is a quantitative risk assessment that we are asked to perform. And it's so important, the toxicological risk assessment, that you will find this word in toxicological risk assessment repeated 55 times in the 1093-18 of 2020. And it's nice that the 1093-18 points to the 1093-17. So as you see here, chemical characterization of a medical device provides the necessary input into the device biological evaluation and toxicological risk assessment. See 1093-1 and 1093-17. So the 1093-18 of 2020 started asking a toxicological risk assessment according to the 1093-17. You can remember that in 2020, the 1093-18 started asking this toxicological risk assessment according to part 17. But in 2020, the available standard, the available 1093-17 was the 2002 version. So a new version, almost a new standard, 1093-18 in 2020, pointed to a standard published in 2002, so to a very old standard. So a clear question, was the 1093-17 of 2002 ready to support the 1093-18? Short answer, no. At that time, the standard was not able to provide the necessary tool to address the question asked in 1093-18. No, not because the standard was wrong. I mean, the 1093-17 of 2002 was a good standard, but simply because in 1093-17 of 2002, we just had an approach to derive tolerable intake, for non-compounds with available data. Again, nothing wrong, but very partial. So tolerable intake for known compounds. And we need to do more in order to complete the toxicological risk assessment. Probably you are already familiar with the margin of safety. You are already familiar with the way we derive the margin of safety. All these concepts were not described in the 1093-17 of 2002. Anyway, as I said, the approach there was right and is still right, the way we derive tolerable intake. We have added new things with the new 1093-17. But the approach to derive a tolerable intake is still a three-stage process. We'll see some differences later on, but we are always performing those three stages. Find relevant information from the literature, derive uncertainty factor and modification factor, and as a third step, calculate the tolerable intake. Is the way we approach to the starting point. We call it point of departure. The way we approach the toxicological data is different with the new standard. The way we calculate the UF is a little bit different with the new standard. And when we have derived tolerable intake, we do more according to the new standard. But let's see those three steps because they are still fundamental. The first stage is the selection of relevant available toxicological information. According to the previous edition, we were looking for the NOEL, so the non-observable effect level, adverse effect level, or the LOEL. So two clear toxicological value for a known compound. The second stage is the assessment of factor to compensate for uncertainty inherent to the extrapolation of experimental data to the given human situation, and for uncertainty in the human response, UF1 and UF2. And they are still there. We still use UF1 and UF2 also according to the new edition of the standard. In 2002 edition, there was also a UF3 that was a factor to assess the quality of the data, a sort of generic factor to assess the quality of the data. We'll see in a while that UF3 is now called UFN to account for more uncertainty. So we are more precise in the uncertainty factor that we use according to the new standard. The last stage of the process is the actual calculation of the tolerable intake. The NOEL is divided by the modification factor to obtain the tolerable intake, assuming, in short, that human beings are more sensitive than test animals. This approach, as I said, is still valid, but it's only a part of the entire toxicological risk assessment. We need to do more, and for this reason and other reason, the 1093-17 was updated. And as I said, in September 2023, so in September of the last year, we have this new 1083-17. Let's see the scope of the standard and the main changes in the document. The scope says that this revised edition specifies the process and requirements for the toxicological risk assessment of a medical device constituent. So this standard provides the tools to address the question that we have seen from part 18. To identify the hazard associated with a medical device, the scope is also to estimate and evaluate the associated risk and control those risks and monitor the effectiveness of the control. So we are here with a standard able to help us in performing a quantitative risk assessment for our constituent. So it's a clear statement in the scope, so we have a clear scope, and the scope is fit for the purpose. We have, of course, some limitation. The standard it cannot be used for everything. The process described in the document is not applicable to the biological risk associated with the physical interaction of the medical device with the body, provided that the chemical exposure is not changed. So this standard is not for a mechanical or physical interaction between, for example, an hip implant and the tissue. Is for the chemicals released from this hip implant. The standard is not applicable for active pharmaceutical ingredients of a device-drug combination product or biological components of the device. So we are not assessing the active pharmaceutical ingredient in a combination product with the standard, but we can assess the compounds released from the device. The standard is not intended to be applicable for marketed medical device that meets the requirements of previous edition of these documents, of course, subjected to justification. So there is no need to reassess the device that
Paolo, you are on mute again.
Microphone is muted.
Don't know what's happening here. Sorry. It seems that as soon as new people are joining the call, the system mute everybody. So sorry for that. I cannot. I hope that I can now see the bar here with the mic, and in case I see it muted, I will unmute me again. So I was saying that the standard is not applicable to everything. Just to summarize, it's not applicable to the biological risk associated with the physical interaction of the medical device and the body. I say there's example the physical interaction between an hip prosthesis and the surrounding tissue, but can be used to address the chemicals, the constituent released from this hip implant. It's not intended, the standard, to be used for a toxicological risk assessment of pharmaceutical ingredient of a combination product, but can be used to assess the constituent released by the device part, and the standard doesn't require to perform an assessment or redo an assessment of devices that are already on the market, devices that were considered to be safe for the intended use according to the previous edition of the standard. So there is no need to redo the assessment, provided, of course, that there are no changes in the medical device. So what's new in the standard? Just to remind you that new clauses were added, and you see here that the clause 5 that is probably the most relevant one, that is toxicological risk assessment within the biological process, was added. This is the main clause describing the entire process. We have a new clause 6, constituent toxicological information, and the way we look for toxicological information. It's not, to be honest, well described the process on how we perform a literature review of the constituent, but at least state the way that we are needed to perform or to look for toxicological information. There is the new clause 7, toxicological contact level, TCL, tolerable intake, and threshold of toxicological concern. As you may know, the TTC concept was developed by ISO TC 194 using the same approach as ICH M7, and was described in a standard not named 1093, is 21726. So it's a standard always published by the ISO TC 194, but not 1093-something. And now we have here a clear reference on how we use the TTC, and we also see later on the toxicological screening. The toxicological screening that is described again in the new part of the standard. We have a clause 8 new about the exposure dose estimation. As we have seen at the beginning, the dose is crucial for us and is crucial for our assessment. And last but not least, we have a clause dedicated to the way we calculate the margin of safety, and how we can judge the margin of safety. So a new clear scope, new clauses to guide us in the process. The process, in short, is the one that you can see here in the figure. We see later on the official, let me say in this way, flow chart provided by the standard. But in short, you see here the different step. We start with the identification of constituent according to part 18, as we have seen at the beginning. We perform an hazard identification, so we identify the constituent of toxicological concern thanks to the qualitative information coming from the chemical characterization. We have, of course, an exposure assessment. We estimate the maximum exposure dose the patient is exposed. This is done thanks to the quantitative information coming from the chemical characterization, generally speaking, coming from an extractable study or a leachable study. As you know, in the extractable study, we have a list of compounds with the quantity released by the device, the quantity of those compounds released by those devices. And we do this exercise, so the hazard identification and the exposure assessment, and the next step for every constituent that is released from the medical device. One by one, we need to identify the constituent from a toxicological point of view. We need to determine the dose the patient is exposed to, and we need to derive the tolerable intake for this specific constituent. Of course, we can use some screening tools we see later on, so we can get rid of some compounds that are released from a medical device. But generally speaking, one by one, we need to identify in the chemical characterization. We need to look for toxicological information in this step. We need to derive a dose for the patient, starting from the data coming from the chemical characterization. We need to derive a tolerable intake for that constituent. And last but not least, probably the most relevant step is the risk characterization, so the assessment of the margin of safety. That is a value. That's why I call it this a quantitative toxicological risk assessment. So let's see the different a little bit more details of the different step of this process. As I said, we need to identify the hazard, and the hazard identification is the process used to evaluate the inner property of a chemical constituent to elicit or to induce one or more adverse health effect in humans. So we are interested mainly in humans. Probably we derive these effects from animal study, but our goal is to protect the patient. And we need also to understand the condition, the route of exposure, the duration, the frequency, the gender, and age necessary for the chemical constituent to elicit this effect. So really, we need to identify all the hazard associated to that specific constituent, and that is the first step. We need information. We need toxicological information in order to go ahead in our assessment. What we are looking for. We are looking for data that can help us in assessing the effects caused by the device. We look for effects in terms of irritation, sensitization, probably if it's available, local tissue toxicity, hemocompatibility. But those effects are relevant. I'm not saying that they are not relevant, but usually we know that those effects are addressed later on by biological testing. We are, let me say, more interested in this data: systemic toxicity, acute, subacute, subchronic, and chronic systemic toxicity. We look for genotoxicity, carcinogenicity data, reproductive and developmental toxicity, because we are able, thanks to those data, to perform the toxicological risk assessment and hopefully waive those biological tests for my device. Let me be very pragmatical, using the same example that I did before, the hip prosthesis. Let's think about an hip prosthesis. This is an implantable device in contact for sure for more than 30 days with the body. If you think about the biological effects to be addressed in your biological information, you will find that more or less all the biological effects are relevant for this kind of device. Probably hemocompatibility is not relevant. And as we know, the chemical characterization is the prerequisite for the entire evaluation. How we approach the entire evaluation. For sure, we need to obtain chemical information, either through the use of existing data or maybe generating new data. Let's say that we perform an extractable study, and the chemical characterization is at the end providing us a qualitative information about the compounds that can be released from the device and the quantitative information about those compounds. As a next step, we perform a toxicological risk assessment on those compounds, and we are mainly interested in systemic toxicity data, in genotoxicity data, in carcinogenicity data, and probably reproductive toxicity data is not relevant for this device. We look for this data, we perform a toxicological risk assessment, and if everything is fine, we can say that we have enough information to address those effects. So there is no need to perform systemic test for this device. There is no need probably to perform genotoxicity test or carcinogenicity test. We waive those tests thanks to the chemical characterization and thanks to the toxicological risk assessment. It's more difficult to address sensitization or implantation effect thanks to the chemical characterization and toxicological risk assessment. Probably implantation effects is not possible at all. But in short, we look for toxicological data. Then it seems a next step, but in the majority of the case is done in parallel. We assess the exposure towards the patient. The exposure assessment is a process used to estimate the quantity of a chemical constituent of toxicological concern that contacts the external dose or enter the internal, it's called the internal dose, the body. So in short, we need to understand the quantity of constituent the patient is exposed to. The process for performing for the assessment is the dose response assessment. It's a process used to extrapolate and identify the point of departure for a chemical constituent of toxicological concern to a toxicological threshold. So the dose response assessment is the way we perform the actual assessment of tolerable intake for the specific compound. The main difference here with the previous edition of the standard is that we are now considering a point of departure, not just a NOEL or a LOEL. This means that we can use any toxicological relevant data other than the NOEL to derive ATE. Of course, in case the NOEL is available, we will go for the NOEL. But a point of departure can be a data derived from a read-across, for example, or a data derived from an in silico method, or any other toxicological data that are relevant for the constituent under assessment. So we are not stick to the NOEL. We are more flexible thanks to the according to the new standard. But anyway, we start with the point of departure, and then we derive a tolerable intake. How we derive a tolerable intake? As for the previous edition, we derive a tolerable intake just dividing the point of departure by modifying factor. And you see here that we are still using UF1 by default is 10, UF2 again by default is 10, and UFN instead of UF3. So the new edition of the standard includes this UFN. Why we using UFN, and what are the value for UFN? UFN is a factor that takes into account additionally uncertainty related to the specific compound. We can take into account the route-to-route extrapolation. Let's imagine that we have data available for one specific route, and we are performing a risk assessment for a device where the patient is exposed according to a different route of exposure. We can perform this extrapolation. We are considering uncertainty related to the exposure duration. We are considering uncertainty in relation to the specific point of departure. We are considering uncertainty in relation to the data applicability. I said a data coming from the read-across. Probably I use an analog, and so I can take into account this uncertainty. And last but not least, as in the previous edition of the standard, we are asked to take into account the quality of the data we used to perform our toxicological risk assessment. But the process is the same. From a point of departure, we derive at the end of the day a tolerable intake. And how we use this tolerable intake and how we use the dose the patient is exposed to, we use the dose in order to calculate, in order to assess the margin of safety. The margin of safety, by definition, is the ratio of the constituent tolerable intake as numerator and exposure dose, the denominator. So it's a ratio between the tolerable intake and the exposure dose. And as you can easily understand, in case the margin of safety is higher than 1, this meaning the tolerable intake is higher than the exposure dose, we can conclude that that specific compound doesn't pose a risk to the patient. And if all the margin of safety, I mean the margin of safety derived for every compound, are above than 1, we can conclude that the risk posed by the medical device is negligible, and we can, as I said before, probably waive some biological tests, mainly the one related to systemic effects, the one related to genotoxicity and carcinogenicity. How and what are the margin of safety that I need to take into account? We need to take into account and we need to calculate the margin of safety according to the exposure, to the period of exposure to the constituent. So according to the duration of exposure to the constituent. We have here 4 different periods described by 1093-17. They are different from the one described in part 1. Probably you are familiar with A, B, and C from part 1, up to 24 hours, up to 30 days, and more than 30 days. Now here we have a little bit of granularity. We have a period that is less or equal 1 day, another period from 2 days up to 30 days. The third one is from 31 days to 1 year, and the fourth one is more than 366 days. According to the specific duration of your device, probably you need to calculate different margin of safety. If you have a device that is less than 1 or less or equal than 1 day, of course you are just interested in acute effects. If the device has a contact that is more than 30 days, you are interested both in acute and subacute effects, and so on and so forth. When you have a permanent device, you are interested in the determination of the margin of safety for acute effects, subacute, subchronic, and chronic effects. You can easily see here how they well correlate with the systemic effects, subacute, subchronic, acute, subacute, subchronic, and chronic effects. The very same effects that we are needed to address in the biological evaluation. Another big new concept was the introduction of the TSL. TSL is really a powerful tool provided by the new standard. Is the toxicological screening limit. The TSL can be used to get rid of some constituent, save the time needed to perform the toxicological risk assessment. The toxicological screening limit is based on the concept of total quantity and the concept of TTC. And the total quantity of a constituent can be used to assess whether the exposure for 1 or more days could result in a cumulative exposure dose that is in a negligible toxicological risk level. And in case we are below the toxicological screening limit, no additional toxicological risk estimation is necessary. So if we are dealing with the total quantity of an extracted compound and the total quantity is below the TSL, there is no need to further assess this compound. Practically speaking, if the compound is below the TSL, there is no need to calculate the tolerable intake for that compound, and there is no need to calculate the exposure dose for that compound, because by default, the margin of safety is higher than 1. We have 2 TSL value. One is 120 microgram, that is for a period of exposure that is less or equal than 30 days. And the other TSL value is 600 microgram, that is for an exposure period that is higher than 30 days. So in case, let's make an example, you have performed an extractable study with the hip implant, and you have estimated that the quantity released by a specific constituent, let's say a palmitic acid, is the total quantity is 100 microgram, you are sure that 100 microgram doesn't pose a toxicological risk. So 100 microgram of palmitic acid is safe because it's below the TSL. You see 100 is below for sure than 120 and is below than 600. So you are on the safe side. There is no need to calculate the tolerable intake for the palmitic acid, and there is no need to calculate the dose the patient is exposed to, because we know that the quantity is below the TSL. What is the total quantity? The total quantity is the quantity coming from usually an exhaustive extraction. From time to time, we can also use exaggerated condition compared to the clinical use. We can even use simulated use extraction, but we need to be sure that the extraction conditions are conservative relative to the intended use of the medical device. When we perform an extraction, an exhaustive extraction, we are sure that we are conservative, and the quantity extracted after an exhaustive extraction can be considered the total quantity, the TQ. It's called TQ, the total quantity. My suggestion is to consider a total quantity when it comes to from an exhaustive condition or a very exaggerated condition. I have seen some pushback from competent authorities considering the total quantity as the quantity coming from a simulated use, just saying that was slightly exceeding the clinical use, slightly exceeding the intended use of device. And you see here how the toxicological screening limit is derived. You have here the link between the three category of part one. So less or equal 1 day, A, B, and C, as you know, and the different toxicological screening limit that can be applicable to the device. Of course, the toxicological screening limit of 120 microgram is for sure the only one, and is for sure applicable for category A, is the only one for sure applicable for category B. In case of category C, we need to apply both. So we have two limits. And if we are between 120 and 600, we can say that long-term, the chronic effects are covered, but the acute one are not covered by this assessment. So we need to calculate the margin of safety just for acute effects. So that's in very short, in few minutes, we can discuss for hours about the what's new in the standard and what are the new tools provided by this standard. I just wanted to highlight the new clause, the overall process, the key new concept, the toxicological screening limit. Now I will share with you a couple of flowchart coming from part 17, describing the process. More or less, it's the same as we have seen at the beginning, and just a couple of additional slides about the way we calculate the exposure dose and the way we calculate the total quantity, because from time to time there is also a need to adjust the total quantity for a specific device. That's the overall approach. We need to establish the medical device intended use according to part one. We need to draft a biological evaluation plan, and of course we need to obtain constituent information according to part 18. So everything we are very familiar. Planning the entire evaluation, performing a chemical characterization as a process for obtaining chemical information, as this is a prerequisite for the entire evaluation. If we have adequate information coming from part part 18, we are fine. We can go ahead with the toxicological risk assessment. Otherwise, we need to further analyze the device. In case we say that toxicological risk assessment is needed, we can use indeed the part 17 to perform the toxicological risk assessment of the constituent. We can even think that toxicological risk assessment is not needed. There are few cases, for example, device made of very well-known materials coming in contact, for example, with intact skin. We have a quite nice list of materials that doesn't require additional information to perform the entire biological evaluation, so there is no need to perform a specific toxicological risk assessment. We justify it. Probably we have already put it in the biological evaluation plan. We need to justify that there is no need to perform the toxicological risk assessment. As I said, in case of toxicological risk assessment is needed, part 17 is our standard, the standard that we need to follow. We need to obtain constituent toxicological information, and that is what we have already seen. If we are dealing with the total quantity, the TQ, and we can compare the total quantity with the TSL, as we have seen. If the total quantity is below the TSL we can just justify, and that's fine. Again, constituent by constituent. In case the total quantity exceed the toxicological screening limit, we need to derive the tolerable intake for that specific compound, or apply, in case really we cannot find any data, a TTC value for that constituent. We need in parallel to estimate the dose. As we have seen, we need to derive the margin of safety. And when the margin of safety is calculated, we need to apply the acceptance criteria. As I said, in case the margin of safety is higher than 1, we are on the safe side. The compound doesn't pose a relevant toxicological risk. In case all the criteria are met, again, we are done. We need just to write our justification. In case some compounds exceed the acceptance criteria, so the margin of safety is lower than 1, we need to do something more, further analyze, evaluate, or control the risk with the link to part 1, and of course our main standard related to the risk management, the 14971. Just another flow chart that is provided in the 1093 part 17 is more clear about the specific approach on the way we calculate or we address the margin of safety. Nothing more than what we have already seen. To be honest, I always said tolerable intake. Just once, probably, I said TCL, tolerable contact level. There is also the possibility to address irritation in a toxicological risk assessment using the TCL concept instead of the tolerable intake. The application of the toxicological risk assessment to address the irritation is very, very limited. My suggestion is, in case you need to address this endpoint for your device, it's better, it's easier, it's even cheaper and faster to address this endpoint through a biological test, rather than using the toxicological risk assessment for the irritation effects. But it's possible, and as you see here in this part of the flow chart, you have also the possibility to apply toxicological risk assessment to address these effects. So the last two concepts. One is related to the total quantity. I say that always that the total quantity is amount of a constituent that is present or can be extracted from a medical device. That's I have to say it's the right definition. But when we are dealing with the total quantity, it's better for us to understand that we are interested in the quantity the patient is exposed to. Let's make a very simple example with the hip prosthesis. I say that the quantity extracted using an exhaustive extraction is the total quantity. Let's say 100 microgram of palmitic acid after an exhaustive extraction. That's the total quantity extracted from the device. But the patient, in the worst case scenario, is not exposed to 100 microgram extracted from any prosthesis. In the worst case scenario, so the total quantity of exposure is 200. Because in the worst case scenario, the patient is exposed to 2 hip implants. So for us, it's important to address or to calculate the total quantity the patient is exposed to. Let's make another example just to add more clarity on this concept and why we are using the so-called SF, so SF value. To take into account the number of the quantity of the medical device that contains the body and the quantity or the number of medical device used in the extraction study. Let's do a different example. For any reason, you have performed an extractable study for a device of, let me say, 100 centimeters square. Because otherwise it was not possible to test because of some limitation in the test system for any reason. Probably even because at that time only that specific size was available for test. That's fine. If you have tested that device, the total quantity is the total quantity extracted from 100 centimeters square of surface. In case the patient is exposed to a bigger device or even more than one device, let's say 250 centimeters square of surface, you need to scale the quantity extracted from 100 ml to from 100 centimeters square to a total quantity that is released from 250 centimeters square. So you have a 2.5 SF factor in order to scale the quantity extracted to the quantity the patient is really exposed to. Because again, our assessment is aimed to protect the patient in the clinical during the clinical use of the product. So that's one important thing related to the total quantity. The other one is related to the exposure dose. We need to calculate the maximum value for a specific intended clinical use scenario. So we have seen at the beginning that we are interested in acute effects, subacute, subchronic, and chronic effects. Of course, according to the duration of body contact between the device and the patient, and we need to obtain information from chemical information according to part 18. Now we have a clear question: is the kinetic the release kinetic available? So we know the way the device is going to release compound or not. If we know the release kinetic, the exposure dose, so the dose the patient is exposed to, is based on this kinetic of release. we need to take into account the worst case, but we have this kinetic. In case we have designed the study without taking into account the release kinetic, that's fine. That's fine anyway. But we are asked to calculate a sort of theoretical release of this compound. So we are basing our evaluation not on the actual release kinetic, but we are performing our assessment based on a theoretical release kinetic. And in any case, we needed to assume the worst case release, and in both cases, we need to perform a worst case. We need to perform an assessment, sorry, in a worst case scenario. So that is it's quite clear. And you see here the different formulas that you can apply in order to determine the dose the patient is exposed to. There is no need to enter into the details of those two value. Just to those two formulas. Sorry, just to highlight one thing. When you are dealing with a release kinetic, you are dealing with quantity released in different period. While, as I said, when you are dealing with data without any clue or any information about the kinetic of release, you need to assume or you need to have a theoretical kinetic in any case. And that's why you divide the total quantity by the number, the assumed worst case release duration, the number of days, the lowest number of medical device exposure days you are needed. To be clear, if you are dealing with the total quantity extracted by device, a device in long-term contact with the patient, where you are more than 1 year, for example, where you are asked to address acute effects, subacute, subchronic, and chronic effects, you are dealing with the total quantity. The total quantity is divided by 1 to address the acute effects. The total quantity is divided by 2 to address the subacute effects. The total quantity is divided by 30 to address the subchronic effects. The total quantity is divided by 365 to address the chronic effects, because it's the lowest number of medical device exposure day for that period. And you end up here in this case with 4 margin of safety that needs to be addressed. Of course, you have 4 exposure dose, but you can, in theory, also dealing with 4 different tolerable intake. Why you are dealing with 4 different tolerable intake? Because probably your point of departure for acute effects could be different from the point of departure for subacute, for subchronic, or chronic effects. So it's really a burden to perform a toxicological risk assessment according to this new standard, and it seems really burdensome performing this kind of evaluation. But what we can use, as I said before, in case we are dealing with the total quantity, we can use the TSL concept and screen the total quantity according to the TSL value provided by the standard. And if the total quantity, of course, the actual total quantity, so the total quantity the patient is exposed to, as we have seen in the previous slide, compare the total quantity with TSL, and if we are below the TSL, we are done, and there is no need to further proceed with the assessment. So I know that it could be a little bit complex. I hope that I was able to provide a short overview. It's just 1 hour and 10 minutes. You know, it's a little bit longer than expected, but we have in any case time for your question and for and hoping I'm able to answer to your question. So just to give you let me say an overview of the standard. Just that's it. I have to say from my side. Thanks for joining us today. For me, it was really a pleasure. I see a lot of participants here. Just before Q&A, special thanks to Tunka and the entire team there for organizing this webinar, taking care of all technical aspects, reminding me that I was unmuted from time to time. I will share this slide, then they will be shared. I think also to you. I just invite you to provide feedbacks and comments. You see here my email. Just drop me an email. So we have time now for the Q&A part. If you like to submit your question by email, please do so. And in case there are unanswered question, I will follow up later on by email. So thanks, and yeah, now I can probably also see the chat if there are questions.
Thank you, Paolo. You should be tired.
Gracias, Paolo.
Thanks to you guys. I see some comments in the chat, but sorry, I cannot read it. Okay, no, is FDA saying that they will agree with the scaling approach? Okay, I can read it and I can answer to that. Well, generally speaking, they do agree with the scaling approach. So this concept is accepted by the notified body and by FDA. That's clear. It's of course always a matter to be sure that the data you are using are in any case relevant for your device. I made before some stupid example. For example, if the device is 100 centimeter square and later on you use 2 devices in the patient, you can use a factor 2, and that's clear. This is accepted. Scaling it can be a little bit more tricky when you are dealing with different sides of your device, because beside the scaling, so to justify the fact that the data coming from another device are applicable to this new device. Again, for example, you are saying, okay, I have performed an extractable study with this size, and then I have a bigger one. But maybe that the ratio between the different parts is not the same as a smaller one. So here is the case to justify that even with a different ratio between the part, you are the data are still relevant. So generally speaking, yes. The short answer is yes, but please be sure that the data are applicable also in the other case. Okay, there is another question. My question is that we'll have a chemical characterization test done on our product. After test, the expert will write a toxicological evaluation. It's mandatory to have a European registered toxicologist for expert for this report. Good question. Again, short one is no. The standard doesn't require to have a ERT or DABT, so a diplomated American Board of Toxicologists, or a recognized individual. For sure, there is there is a request to have people involved in the assessment with the right knowledge and the right expertise to perform this assessment. For sure, somewhere in your biological evaluation plan, in your toxicological risk assessment, you need to identify the people involved in the assessment. Usually, it's not a single-man show. Let me say in this way, it's a team effort where there is the manufacturer, there is an external expert. And what is needed is somewhere in your quality system, in the technical file, in the report provided by a third party, you have the demonstration that people involved were knowledgeable to perform that assessment. Again, no need to use a European registered toxicologist. There is a need to use, let me say this, even if it's not, we are not using people, to use an expert or people with a knowledge of doing that. By the way, there are European registered toxicologists that are really nice, but they are coming from a completely different field. You can be a ERT for a forensic toxicology, and they are ERT, but probably they have no clue on how to perform a toxicological risk assessment. I'm an ERT. If I'm asked by a court to provide an expert opinion on something related on forensic, I'm not the right one at all. I have no clue on how this is done, on how we perform this kind of assessment. So if in your quality system you decide that a ERT is needed, that's fine. It's your decision. Look for an ERT with the knowledge in medical device. Are specific database or sources for TCL value? Well, that's a good question. There are databases. There are several sources. I can provide a list of database that can be used to Tunca, and it can provide it to you. For sure, thanks to some effort in Europe from ECA, from OECD, we have several databases available. We can use also value derived for pharma, for the pharmaceutical domain. So there are databases, as I said, OECD database, ECA database, and so on, so forth. Also, FDA has some database, for example, for some colorants that are used in different medical device application. I can provide you with a list of these database by email.
Paolo, there is another part of this question, which is part 1.
Okay, sorry. Okay, I missed the sorry. I was just scrolling. I went just directly from part 2. Sorry for that. Okay, part 1. From the scientific and regulatory perspective, we can use TCL approach to justify irritation, or it's mandatory to perform biological test to address irritation endpoint. Oh, that's a good really a good one. From a scientific point of view, I have to say that TCL is based on TTC, and TTC, at least the value that are described in ISO TC 21726 are, that by the way, are the same as ICH M7, so the very same as pharma. Those values are derived for systemic effects and genotoxicity and carcinogenicity. So they are the TTC value. They are not intended to cover the irritation part. And as long as they are used, the TTC value, to derive the TCL, the toxicological the toxicological screening value, they are not they do not cover. So that is for the screening level. For the TCL, so the tolerable contact level, it's appropriate to use this tool, the toxicological risk assessment, to address irritation. So that's fine. I have to say that from a scientific and a regulatory point of view, yes, you can do that. It's for me, it's a practical question. On one end, when you are dealing with the device required to be assessed for cytotoxicity, irritation, and sensitization only, probably there is no need to perform an extractable study and then use the data coming from an extractable study to perform a toxicological risk assessment just to justify the irritation. We have in Europe an in vitro method, so we are fine just to perform it. It's really a practical approach. And my second consideration about that, when you are dealing with a very complex complex device, you have probably you are dealing with a lot of compound, and it's not always the case that you find relevant data for irritation for some compounds, and in a lot of cases you don't have data, so you are using probably TTC value, or you derive this value using an in silico approach or a read-across approach, and so on and so forth. And it's not always the case that you are able to address irritation, or you are able to find relevant data to calculate the TCL value. So again, from a regulatory and scientific point of view, it's fine, but we have some practical limitation in performing that. There is any guidance available for how to design a kinetic release study? Again, short answer, no. We have guidance. We have guidance for degradation study, but not for kinetic release. It's more a matter to design your own your study to address this kinetic. Generally speaking, part 18 is providing a nice guidance on the selection of solvent. It's providing guidance on how to select the temperature, how to select the condition, but the release kinetic is more up to the to your design. Usually, in my experience, I have seen at least to consider the 24 hours endpoint in order to have an idea of the acute effects, and probably add a second time point after 48 or 72 hours, and eventually a third one, and then combine this data with the with also a total quantity extraction or exhaustivity extraction. Again, being very pragmatic, designing or let me say the effort associated to a full kinetic study is quite high, both in terms of time and in terms of budget needed for this kind of study. It's possible to address material-mediated paragenicity with chemical characterization and toxicological risk assessment. No, not because it's not possible from a theoretical, from a scientific point of view, but because we have really there are almost no data for the majority of compound about material-mediated paragenicity. I have to say that material-mediated paragenicity was introduced in part 1 in 2018, and we are going to remove these from the endpoints to be evaluated for every device during the revision of part 1, because we know that material-mediated paragenicity is really rare, and we have few compounds causing a material-mediated parogenicity, and the majority of them are not associated with a medical device. Lysergic acid, cocaine, for example, are causing a parogenicity, and it's a chemically mediated effects. So you can use probably you can support your overall risk assessment about the fact that there is material-mediated parogenicity is not relevant for the device, just using qualitative information coming from the toxicological risk assessment, but it's not, let me say, done in a quantitative way. It's sufficient the margin of safety is greater than 1. Well, it's equally conservative if the input of Morse aren't 1, 100, or 1000. That's again another good point. I say that it's enough that the margin of safety is greater than 1. There is a caveat, of course, provided that both the numerator, so the tolerable intake, and the denominator, the quantity, the dose, are conservative. So in case you are you are performing this assessment and you are deriving the margin of safety in a very conservative way, higher than 1 is enough. Of course, when you have more than 1, more than 10, more than 100, you are more than happy on that. So no need to reach 10, 100, and so on and so forth. I have to say that FDA it's a little bit more strict on that. They say that a margin of safety higher than 10 is considered to be protective. So they have raised the bar up to 10, and they from time to time need even some justification when you are between 1 and 10. Again, because of the fact that you need to be sure that the factor used for margin of safety are conservative enough. If understood correctly, you have mentioned that it is not possible to address implantation endpoint with chemical characterization and toxicological risk assessment. Yes. Okay, I got your point. I know that from the categorization of some device, local effects after implantation seems or are required even for devices that are not intended to be implanted. You are perfectly right. Usually effects, local effects after implantation are a combination both of physical feature and the chemical release. So I say that implantation study is or toxicological risk assessment is not used in lieu of implantation study because of this double nature of the implantation study. I'm thinking, for example, when you are dealing with a substance-based medical device that is encoded with breached or compromised surface, for example, a cream or a gel that is encoded with breached skin, with a wound. In that case, probably, and you are asked to address local effects after implantation. The implantation study is not so relevant, and you can in some way use chemical information and toxicological risk assessment to address also local effects. And of course, beside that, you have the intracutaneous activities, and that's the overall approach that you need to design in your biological evaluation plan. I know that we are a little bit ahead, but could you talk a little bit for the reportation of toxicological risk assessment design and standard? Okay. Well, just in short, you need to provide to the competent authority or a report with at least three different this different aspect. So that there is for sure a clear part related to the toxicological information for the compounds. You should find a clear part on the evaluation of the dose the patient is exposed to, and another clear part about the margin of safety for every compound, and of course the conclusion of the assessment. So generally speaking, there is the definition of the medical device. There is a section related to the input data, the data coming from an extractable study, and the rest of the assessment. The very last one, well, chemical characterization. Just to be clear, chemical characterization is needed for every device, but chemical characterization doesn't mean chemical test. So that's the main. Chemical characterization is a process for obtaining chemical information. Let's make a very simple example. You are dealing with a device with intact skin. You are dealing with a very very well-known materials. You have chemical information because you know the composition of the materials. You have safety data sheet. You have information about that material. That's fine. That's enough for the chemical characterization. There is probably no need to generate further chemical data for that specific one. Is the local toxicity risk assessment of extractable lesion also required for contact outside the dermal route? No. In case we are dealing with the systemic exposure, we are not so interested in the TCL. That's why I focus my presentation on TE rather than TCL. If you are dealing for an implant, there is no need to assess the TCL. Just to be clear. Okay, now I think that I am over. Sorry for being late, but thanks for your question. It seems that you are passionate like me about this standard. Thanks for being so many here.
Thank you very much, Paolo. Again, hope to see you in 2025 with new topics.
Yeah. Sure about that. Thanks for your time. It was a pleasure, and see you soon for sure. Bye bye.
Bye bye.
Take care.
Thank you,
Bye
Paolo.
bye. Thank you. Bye.
Tuğrul Bey, siz kapanışı yapabilirsiniz.
Teşekkür ediyorum sağladığınız bu imkan için. Bu kadar kalabalık insanı, katılımcıyı da yönetmek gerçekten önemli bizler için. İSEK Akademi'yi ve İSEK Derneği'ni takip etmeye devam edin. Bu tür etkinliklerimiz zaman içerisinde oluyor. Çok da kıymetli olduğunu görüyoruz bu gelen sorulardan da. Çok teknik, derin teknik bir konu olmasına rağmen hem katılımcı sayısı olarak hem de sorulan soruların niteliği olarak gerçekten kıymetliydi bizler için. Umuyorum sizler de umduğunuzu buldunuz, sorularınıza yanıtları net olarak aldınız. Ben öyle gördüm en azından. İSE'yi takip etmeye devam edelim. Hatta mümkünse derneğimize üye olalım ki ekstra avantajlarından da ve network'ün içerisinde bulunmak için de faydalanmış olalım. Çok teşekkür ediyorum Adikuale'de bu toplantıyı düzenlediğiniz için. Görüşmek dileğiyle. Hoşça kalın.
Rica ederiz. Görüşmek üzere.